So nucleic acids are our final class of biomolecule. And we're principally going to be talking about DNA, but there is RNA as well. DNA stands for deoxyribonucleic acid. And deoxyribonucleic acid is a recipe book for protein. So all of the proteins in your body were originally, the order of amino acids was originally specified by your DNA in every single one of your cells. So DNA, it comes in these little packets called genes, and genes say, here is the order of amino acids that you need to put into a particular protein. So there is DNA, which lives in the nucleus of your cells, and then there is RNA, which stands for ribonucleic acid. The difference being that DNA is deoxyribo, and RNA is just ribonucleic acid. And these are chemical codes. This is a chemical language that allows your cells to specify which amino acids get made, and RNA helps with this process. And we're going to learn all about RNA later on in the semester. So here is the double helical shape of a strand of DNA. So you've got your bases here. Each piece of DNA is a base pair. There are A's, G's, T's, and C's. And you can see there are complementary base pairs. So one base pair sort of fits with another. T's go with A's always. C's go with G's always. So anytime you have a C on one side, you have a G on the other, and vice versa. So anytime you have G on one side, you will also have a C on the other. Same with A's and T's. So C's go with G's, A's go with T's. Okay, and like I said, they're sort of this staircase double helix shape that DNA takes on. So nucleic acids are a polymer, and remember, a polymer is just a repeating series of monomers, and each monomer subunit, or each nucleotide as it's also called for DNA, consists of sugar, phosphate, and one base. So A's, G's, T's, or C's. But in RNA, there are no T's. These are replaced by uracil. A stands for adenosine, or adenine, by the way. C stands for cytosine. G stands for guanine, and T stands for thymine. And then U stands for uracil. But the only time you see uracil is in RNA. When you're trying to make a strand of DNA, you use the same dehydration synthesis we've seen before, where those two monomers are going to get together and a water molecule is going to be released from them. So again, this is just this is maybe a little bit much, actually, but this is just showing how the chemical structure of each piece of the DNA, and it's showing that this phosphate actually represents an actual phosphate group out here with 3 oxygens and the pentose sugar. I'm probably not going to have you guys draw like in this much detail. I may show you in lab a slightly more simplified version of DNA that I'm going to want you to draw. There are of the various bases, there are 2 different types. There are these things called pyrimidines, which are these single ring base pairs. And there are these things called purines, which are these double ring base pairs. So these purines are, you can see physically, a little bit larger. They have kind of the same size, only they've got this extra ring in addition. So the way I always remembered this in biochemistry was pure as gold for purines. So pure from the purine, as gold for Ag, and then pi cut for the pyrimidines. So PYCUT are those pyrimidines. I won't ask you for the shapes of these or anything like that, but I might ask you, hey, what's a purine? What's a pyrimidine? What are the differences? What are the similarities? So this is just showing the process of DNA replication, because as we're going to discover later on, every single time your cell divides, it needs to make a copy of its DNA so that the new cell can have DNA as well, and that's kind of where your DNA comes from. So this is just showing you the double helix. When it's trying to copy itself, the strands actually separate out, and then you have some enzymes. Remember, enzymes are proteins. Those enzymes are going to come in and rebuild the staircase until you have 2 identical strands at either end. That's just kind of like a preview for later on this summer. So one strand of DNA has hundreds to thousands of genes. So the DNA double helix has 2 strands on it. They are bonded together by hydrogen bond, and like I was saying earlier, A pairs with T, T pairs with A, C pairs with G, and G pairs with C. DNA is a single strand of nucleotides, and actually what happens is your DNA has a certain code that it's specifying, and whenever your body needs to access that code, it goes and makes an RNA copy. Goes and it writes down, if you will, makes a little copy of the information it needs. DNA also needs to replicate itself. The strands basically separate, and the new complementary nucleotides will join in the process of mitosis. And here is, this is a little phylogenetic tree that is showing you the relationships among these species. So here are human beings, and you can see they're quite closely related to gorillas, and they're still pretty closely related to gibbons and rhesus monkeys, but not as close. And it's showing back here a common ancestor. So what this is saying is, for one particular gene, hemoglobin in this case, the difference in the gene for human hemoglobin and gorilla hemoglobin is 1 amino acid. 1, which is pretty small if you think about, amino acids are these 200 or so, usually more than 200 amino acids all hooked together to make a protein. So the hemoglobin protein between humans and gorillas is 1 base pair off, or not base pair, 1 amino acid off. And between humans and gibbons, it's 2 amino acids off. So these 3 organisms are incredibly close to one another, at least in terms of this one particular protein. And then rhesus monkeys, they're still pretty close, but they're further out. When you get out to mice, they're only 27 amino acids apart, which is still really quite close. And then you consider, frogs are 67 base pairs different, which you might think is incredibly different, and I keep saying base pairs, 67 amino acids off, which you might think is incredibly different, but it's really not all that different in the grand scheme. So it's just showing you there's this evolutionary thing that can be tracked using DNA.